Welcome to our bite-sized session on unlocking molecular mysteries in T-level science. Today we will delve into the intricate world of molecular biology with this comprehensive content buster tailored for T-level science. Explore the complexities of meiosis, protein structure, amino acids and chromatography, essential topics for understanding the molecular puzzle of life. So today we embark on a journey into the microscopic world of molecules. Together we'll unravel the mysteries of meiosis, which is understanding genetic variation through meiosis is fundamental. Protein structure, so how amino acids shape protein function and structure. Chromatography, applications, how versatile chromatography techniques have diverse industry applications. We'll explore experimental skills through an example of hands-on activity, develop practical and analytical skills. We'll explore interdisciplinary connections, so how T-level science integrates biology, chemistry, and analytical techniques. And we'll explore real-world relevance, how concepts are applicable in pharmaceuticals, biotechnology, and environmental science. Meiosis is not just another form of cell division. Meiosis is a specialized form of cell division crucial for producing genetically unique gametes used in sexual reproduction. It involves 2 rounds of division, resulting in 4 daughter cells with half the number of chromosomes as the parent cells. During the meiosis 1, genetic variation occurs through crossing over, where homologous chromosomes exchange genetic materials. In box A, the cell undergoes replication of DNA. In box B, the cell completes meiosis 1 and the chromosomes number is halved and the process of crossing over takes place. And in box C, the cells complete meiosis 2. Purpose is to produce 4 haploid cells known as gametes. So meiosis produces 2 daughter cells with the same number of chromosomes as the parent cell, essential for growth and tissue repair. Meiosis, on the other hand, generates genetically diverse gametes, crucial for sexual reproduction. The 2 stages of meiosis ensure genetic variation offspring, a vital aspect of evolutionary adaptation. Proteins are the workhorses of life, performing an array of functions from catalyzing chemical reactions to providing structural support. Understanding their structure and function is key to unlocking the mysteries of life itself. Proteins are essential macromolecules with diverse functions in the body. Amino acids are the building blocks of proteins, with each amino acid differing in its side chain. The structure of a protein determines its function, with variations in shape influencing its role in biological processes. Amino acids may seem like simple molecules, but their arrangement holds the blueprint for life. Each amino acid contributes uniquely to the complex machinery of protein synthesis, shaping the biological landscape. Amino acids are composed of an amino group, a carboxyl group, and a unique side chain. The sequence of amino acids in a protein is dictated by the genetic code encoded in DNA. Different combinations of amino acids give rise to a vast array of proteins with diverse functions. So how do these concepts interconnect? Meiosis, protein proficiency, and amino acids are interconnected through their roles in the fundamental processes that govern life. Here's how they connect. Meiosis, as a process of cell division in sexually reproducing organisms, introduces genetic variation through mechanisms like crossing over and independent assortment. This variation ensures that offspring inherit a unique combination of genetic material from their parents. A diverse gene pool provides the raw material for the synthesis of a wide array of proteins, each with its own unique structure and function. Amino acid synthesis. So amino acids, the building blocks of proteins, are synthesized in cells through complex biochemical pathways. The genetic information encoded in DNA determines the sequence of amino acids and protein. During meiosis, genetic recombination and crossover events shuffle genetic material between homologous chromosomes, leading to the creation of genetically diverse gametes. These gametes, upon fertilization, contribute to the genetic diversity of offspring, ultimately influencing the variations in protein sequences and functions. Gene regulation, expression regulation. So meiosis also plays a role in regulating gene expression patterns. The process of transcription and translation, which ultimately leads to the synthesis of proteins from mRNA templates, is tightly regulated during meiosis. This regulation ensures that specific proteins are produced at specific times and in specific cell types, contributing to the overall function and development of an organism. So, impact on phenotypic traits. The genetic diversity generated through meiosis can influence phenotypic traits, including those related to protein structure and function. Variations in amino acid sequences resulting from genetic recombination events during meiosis can lead to differences in protein folding, stability, and activity. These differences in protein properties can in turn impact an organism's phenotype, including appearance, behavior, and physiological characteristics. So, in summary, meiosis, protein proficiency, and amino acids are interconnected through their roles in generating genetic diversity, regulating gene expression, and influencing protein structure and function. Understanding these connections provides insights into the complex relationship between genetic inheritance, molecular biology, and organismal traits. So now let's explore chromatography. It is like a molecular detective, separating mixtures into their individual components for closer inspection. From crime scene investigation to environmental monitoring, chromatography plays a vital role in unraveling scientific mysteries. You have thin layer chromatography or TLC. So the principle of TLC separates components of a mixture based on their affinity for mobile or solvent or stationary phase coated plate. Components with a higher affinity for the mobile phase move faster, while those with a higher affinity for the stationary phase move slower. TLC is used to detect the number of components present in a mixture and assess their purity. This is achieved by visualizing the separated components under UV light or by using chemical reagents that react with specific compounds. TLC is widely used in various industries for qualitative analysis, such as pharmaceuticals to monitor reaction progress and in forensics to identify illicit substances. Next, we have column chromatography. In the column chromatography, a mixture is separated as it passes through vertical column, filled with a stationary phase. Components are eluted based on their interaction with the stationary phase, with the least retained component eluting first. Column chromatography is particularly useful for isolating pure compounds from complex mixtures, making it a valuable technique in organic synthesis and natural product isolation. Gas chromatography, or GC. GC separates volatile compounds in a mixture based on their partitioning between a mobile gas phase, so carrier gas, and a stationary liquid or solid phase, coated column. Compounds with high vapor pressure elute faster. GC is widely used for quantitative and qualitative analysis of volatile organic compounds, or VOCs, in environmental, forensic, and pharmaceutical application. It is highly sensitive and can identify trace amounts of compound. And then high performance liquid chromatography, or HPLC. HPLC separates components of a mixture based on their affinity for a mobile liquid phase, or pressurized solvent, and a stationary phase, the packed column. Components are eluted at different times depending on their interactions with the stationary phase. HPLC offers high resolution sensitivity and reproducibility, making it suitable for a wide range of applications, including pharmaceutical analysis, environmental monitoring, and food safety testing. And finally, mass spectrometry, or MS. So the principle of MS is separating ions based on their mass to charge ratio, or MZ, and detects them using a mass analyzer. By measuring the mass of ions, MS can identify molecular ions and fragments present in a sample. MS is a powerful tool for identifying unknown compounds in a mixture, as each compound produces a unique mass spectrum. It is widely used in metabolomics, proteomics, and drug discovery. So JC, HPLC, and MS are not only used for qualitative analysis, but also used for quantifying the amount of each component present in a mixture. This quantitative information is crucial in various fields, such as pharmaceuticals for determining drug concentrations in formulations, and environmental science for assessing pollutant levels in water and air samples. These techniques are essential in analytical chemistry for separating, identifying, and quantifying components in complex mixtures, providing valuable information for various scientific and industrial applications. So how do these concepts interconnect? So the connection between meiosis, protein proficiency, amino acids, and chromatography lies in their collective contribution to understanding and analysing the molecular components of living organisms. Here's how they link together. Meiosis generates diversity, resulting in variations in protein sequences among individuals. These diverse proteins play crucial roles in various cellular processes and physiological functions. Chromatography techniques such as high performance liquid chromatography, HPLC, can be employed to separate and analyze these proteins based on their unique properties, including size, charge, and affinity for certain molecules. In relation to amino acid analysis, amino acids, the building blocks of protein, exhibit distinct proteins that can be exploited in chromatography. Techniques like TLC and column chromatography can separate and identify individual amino acids in a mixture. This analysis provides insight into the composition and abundance of amino acids, which are essential for understanding protein structure and function. So protein structure and function. So chromatography techniques are instrumental in studying protein structure and function. For example, chromatography can be used to determine the quaternary structure of proteins by analyzing their interactions with specific ligands and antibodies. Additionally, chromatography can help elucidate the role of post-translational modifications, such as phosphorylation, in protein function and regulation. So what are the biological applications? Chromatography has diverse applications in biological research, including protein purification, drug discovery, and biomarker identification. By separating and analysing complex biological samples, chromatography allows scientists to characterize the molecular composition of cells, tissues, and biological fluids. This information is invaluable for understanding disease mechanisms, developing therapeutics, and advancing biomedical research. So in summary, meiosis, protein proficiency, amino acids, and chromatography are interconnected components of molecular biology and analytical chemistry. Together, they facilitate the study of biological processes. the structure, function, or relationship of biomolecules, and the characterization of complex biological systems. Understanding these connections provides a holistic approach to investigating the molecular basis of life and disease. So we're now going to explore an experiment that can be done in the classroom. On the screen will be more detailed instructions, and I will then summarize throughout. So the aim is to separate and analyze ink pen components using chromatography techniques. So the materials will be components such as ink pens of different brands and colors, filter paper, chromatography chamber, solvent system, e.g. water or ethanol, TLC plates, and capillary tubes or pipettes. So the procedure. So starting with paper chromatography. So you can see the full procedure here on the screen. But just to summarise, you're going to prepare filter paper trips and mark a line near one end, spot ink dots on the line and immerse strips in solvent, and allow solvent to migrate and analyse separated components. So again, follow these instructions on the screen fully and in detail to make sure that you're completing the experiment correctly. So that's paper chromatography. But you could also do a thin layer chromatography or TLC. So again, more detailed step-by-step instructions are on the screen. But to summarise, you're going to mark TLC plates and spot ink samples, develop plates in a solvent chamber and visualise components. Calculate RF values for each component. So remember, there always should be safety precautions taken into account, such as handle solvent carefully, work in a well-ventilated area, and wear gloves and goggles. So how does this experiment demonstrate the link to meiosis, protein structure, and chromatography? So in relation to meiosis, the experiment allows students to observe genetic variation, a concept central to meiosis. By analysing the different ink components separated through chromatography, students can understand how genetic diversity arises, similar to the variations introduced during meiosis through processes like crossing over and independent assortment. In relation to protein structure, ink pens contain various pigments and compounds that contribute to their colour. These components represent different proteins and molecules. Through chromatography, students can separate these compounds based on their properties, akin to the separation of amino acids in protein analysis. Understanding how chromatography works provides insight into the techniques used to study protein structure and composition. So in relation to chromatography, the experiment directly involves paper and TLC techniques. By performing these chromatographic separations, students gain hands-on experience with analytical methods commonly used in biochemical research. They learn about the principles of chromatography, such as solvent migration and component separation, which are applicable to various fields, including protein analysis and forensic science. So let's summarize our key takeaways. So meiosis and genetic variation. Understanding the intricacies of meiosis is crucial for comprehending genetic diversity. Meiosis introduces variation through processes like crossing over and independent assortment, shaping offspring characteristics. We've explored protein structure and function. So proteins are essential for various cellular processes and physiological functions. Amino acids, the building blocks of proteins, determine their structure and function. Chromatography techniques provide insights into protein composition and diversity. We've explored chromatography applications. So chromatography is a versatile technique used for separating and analysing mixtures. It finds applications in various industries, including forensic investigation and environmental monitoring. Hands-on experience with chromatography enhances analytical skills and understanding of biological processes. We've explored experimental skills development. Practical activities such as paper and TLC offer students hands-on experience in scientific techniques. Applying scientific knowledge in experiments fosters critical thinking and problem-solving abilities. Linking experiments to theoretical concepts reinforces understanding and promotes active learning. We've also explored interdisciplinary connections. So, T level science integrates concepts from biology, chemistry, and analytical techniques. Understanding the interplay between genetic processes, protein synthesis, and chromatography techniques enhances holistic comprehension and molecular biology. And finally, we've explored real world relevance throughout. Concepts learned in T level science have practical applications in various industries, including pharmaceuticals, biotechnology, and environmental science. By mastering these concepts, students are equipped with skills relevant to the evolving demands of scientific research and industry. So overall, this T-level science content buster provides a comprehensive overview of essential topics, fostering a deeper understanding of molecular biology principles and their real-world implications. Thank you for joining me in this bite-sized session.